We share more than love with our pets. We also share microbes, including antimicrobial resistant or AMR organisms. Researchers are finding that companion animals like dogs and cats are emerging as important sources and sinks of resistant microbes, with implications for the health of pets and pet owners alike. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Now, scientists have known for a while that pets harbor resistant organisms, particularly bacteria. How prevalent are those microbes in companion animals? Well, that's harder to pin down, due to limited surveillance systems that bring the picture into focus. However, there is a growing repertoire of research that offers some insights. For example, a study from China examining the prevalence of AMR among pet-derived E. coli, Klebsiella pneumoniae, and Staphylococcus species, among other bacterial pathogens, from 2000 to 2022 showed increasing rates of resistance over the study period, including for so-called last resort antibiotics like colistin. Another recent study examined strains of non-typhoidal salmonella isolated from domestic dogs between 2017 and 2023. All strains possessed resistance genes for medically important drug classes like aminoglycosides and quinolones. And still another study found that out of 940 bacterial isolates from infections in companion animals, including species that cause human infections like K. pneumoniae and Pseudomonas aeruginosa, nearly 80% were resistant to at least one antibiotic, while almost 45% were considered multi-drug resistant. Although rates of resistance vary between studies, based on these and other analyses, AMR in pets appears to be common, and the prevalence is likely increasing. But what is driving the spread? The reasons mirror those perpetuating AMR in humans. For example, just like healthcare centers are hotspots for AMR infections in people, veterinary clinics are environments where resistant organisms survive and thrive, transmitting to and between pets. The more veterinary care a pet requires, the higher the risk of transmission. Lack of awareness and clarity on antimicrobial stewardship is also part of the problem. When antimicrobials are used indiscriminately, microbes have more opportunities to develop resistance to the drugs. There are guidelines for antimicrobial use in companion animals, but how closely veterinarians are following those guidelines is unclear. Pet owners must also comply with prescribed administration, making sure to give the antibiotic at the right time, at the right dose, and for the right length of time. But broad compliance among pet owners is, again, a bit hazy. What is not hazy is the fact that people are key contributors to the spread of AMR microbes to and between pets. AMR is a One Health issue, shaped not just by animals, people, and the environment, but also interactions between them. Since most of the antibiotics used to treat pets are the same as those used in humans, and that the same microbes can affect both pets and people, the state of AMR in animals has a direct implication for human health, and vice versa. If humans harbor an AMR organism, they can transmit it to their pets directly or indirectly by way of petting, kissing, and simply sharing a household. Pets can, in turn, transmit AMR microbes to humans and/or other animals. This can happen for a range of potentially problematic bacteria. For instance, companion animals infected with methicillin-resistant Staphylococcus aureus, or MRSA, likely pick up the microbe from humans. On the other hand, dog owners infected by methicillin-resistant Staphylococcus pseudointermedius presumably contracted the bacterium from their pet, as S. pseudointermedius is common in dogs and can sometimes cause infections in people. Regardless of the source of transmission, microbes can swap back and forth between a pet, their owner, and other animals or people with whom they engage. AMR genes can similarly be shared between bacteria from different hosts. But how often does such microbial transmission lead to a full-blown infection? Well, that depends. The tricky part about AMR is that it is all about potential. An organism could be harmful, but it doesn't mean it will be. And there are various factors, such as medical care and the immune status of pets and their owners, that determine whether a microbe will transition from simply being present to being a problem. Indeed, a resistant microbe may live harmlessly in the gut of a cat, but trigger a difficult-to-treat infection in their immunocompromised owner. The risk is thus highly individualized. Nevertheless, understanding human-pet AMR transmission can help researchers frame and mitigate the health risks on a broad scale. Gathering this knowledge involves a range of tools that characterize the presence and movement of AMR organisms in and between pets and humans, from antimicrobial susceptibility testing to genomics. From a transmission standpoint, genomic analyses are especially insightful, as they can shed light on similarities between drug-resistant microbial isolates from humans and their pets. If the isolates are genetically identical, that's a good indicator of microbial sharing between pets and people. Epidemiological analyses, which focus on the logistics of spread, help determine directionality of that sharing. Did a pet give a microbe to a human, or did the human give it to the pet? These collective insights create a map of AMR transmission in pets and humans, and ideally, pave the way for the incorporation of companion animals into surveillance efforts. Still, many studies do not examine isolates from both animals and their owners when conducting susceptibility testing and strain typing, which is critical for piecing together the puzzle of AMR transmission in humans and pets. Moreover, antimicrobial susceptibility testing must usually be covered by pet owners. Many owners will opt out of these tests because completing them is not economically desirable or feasible, creating a barrier to collecting those important data. With that in mind, scientists emphasize the need to prioritize gathering more information about AMR in pets and the bidirectional exchange with people. This doesn't mean just more extensive studies, but also greater collaboration between public health departments and veterinary practices. Connecting data from the veterinary industry with those from the human side could influence both veterinary and medical approaches to treating AMR infections. Regularly collecting information on pets and pet owners in both healthcare and veterinary settings could help determine an individual's risk of AMR exposure or help pinpoint the source of an infection. It's also worth noting that while dogs and cats make up a good chunk of the 134 million pets in the US and beyond, they are not the only companion animals out there. Insights into AMR in other pets will also be valuable. While more research is needed, in the meantime, we can work to limit the spread of AMR to and among our animal friends. For instance, veterinary centers should ensure they are following comprehensive infection control measures, strict hygiene practices, and disinfection protocols. Across the veterinary industry, increased education on antimicrobial stewardship guidelines and the impact of AMR for veterinarians and pet owners can help curb the spread. At home, owners can do their part by keeping animals and homes clean, washing their hands after playing with or snuggling with their pets, and minimizing intimate contact, like letting their pet lick their face. Ultimately, it's about loving and caring for our pets, but doing so with microbes in mind. That's all for today's Microbial Minutes. If you enjoyed this video, subscribe to ASM's YouTube channel for more. As always, I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you next time.